Welcome to the deep dive. So if you've been anywhere near modern biology lately, you know the data situation. It's a lot. Single-cell RNA sequencing gives us this just absolute flood of information. Millions of cells, thousands of genes per cell. It's this huge combinatorial storm. And finding the actual signal in all that noise, it's incredibly tough. It feels less like science sometimes and more like, I don't know, trying to understand a city by looking at every single brick individually.
Yeah, it's a massive challenge.
Yeah.
But we finally have, well, maybe not the answer, but a really powerful new tool to approach it. Today we're diving into something called Cell2Sentence. And it's quite radical, really, because it connects that super complex biological data, single-cell gene expression, with arguably the most powerful tool we have right now, large language models. LLMs.
Okay, Cell2Sentence. So it reframes the complexity. It makes it readable for an AI.
Exactly. It turns it into something an LLM can, read, reason about, even write about, almost like it's just another piece of text like scientific literature.
All right, so for you, the listener, our mission today is to really unpack how that works. We want to show you the kind of genius behind the simple encoding idea, get into the details of their huge C2S scale 27B model, and crucially, walk through this validated cancer result that really proved this isn't just theory, it works in actual human cells on the lab bench. Okay, let's start with that core idea then. The whole thing hinges on turning, this messy, high-dimensional cell data into a simple sentence. It sounds almost too simple. What's the exact structure? How does that structure let LLMs understand biology?
Well, it's interesting because the simplicity is the strength here. Fundamentally, a cell sentence is just a list of gene names, separated by spaces. But the key bit is they're ordered by expression level. So most express gene first, then the next all the way down.
Wait, so hang on. You're saying they basically ignore the amount of RNA for each gene, the actual counts that biologists usually obsess over. Isn't it just the ranking? Doesn't throwing away the magnitude loot really important information?
That's a great question. And it really gets to the cleverness of the design, I think. See, with single-cell data, those raw counts, they are super noisy. You get technical variations, batch effects between experiments, even just cell size differences. All that stuff messes with the specific numbers. But the relative ranking, which genes are sort of dominating the cell's activity right then, that hierarchy turns out to be much more stable. It holds up better against all that experimental noise.
Ah, okay. So you trade noisy numbers for a stable structure, a ranked list. And that list, because it's just text, words separated by spaces, that's the bridge that lets the LLMs get involved.
Precisely. And that simple text format, it immediately gives you two huge advantages. First, the efficiency. You can tap into these incredibly powerful existing LLMs like the Gemma 2 family. You use their training setups, which are already built to scale massively. You don't have to reinvent the wheel building custom biology-specific transformers from scratch. You leverage what works.
Right, using something off the shelf like Gemma, but does that lock you in? Any constraints compared to a totally custom model?
Well, the key is they're using the open source Gemma 2 architecture, so there's flexibility there. The main thing is avoiding the need to build the basic sequence processing engine again, which ties into the second benefit, what they call bilingual training.
Bilingual? How so?
Because the input is now sentences, you can train the model on both the cell sentences and enormous amounts of regular biological text. Things scientific papers, lab notes, metadata, even questions in plain English. So the model learns to be fluent in both languages, It understands the literature about gene X, and it understands the expression pattern involving gene X in a cell sentence. And this is crucial for actually using the model later, like asking it questions about the cells. You could literally prompt it, what cell type is this? And feed it the cell sentence.
Okay, that totally changes the training objective then. If it's learning sentences, it must be using that standard predict the next word goal, like GPT and other LLMs, learning to continue the sentence.
That's exactly right. The model learns to predict the next gene in that ranked sequence in a way that makes biological sense based on everything it learned from the text and metadata. And the authors make a point of contrasting this with, say, older methods like Geneformer, which often used mask token prediction like filling in blanks. Cell2Sentence is about generation, predicting what comes next, which is much better suited, they argue, for biological tasks, like predicting how a cell might change after you perturb it with a drug.
So if biology is now language for this model, the proof has to be in the scaling, right? Does it follow those same scaling laws we see with language models? Bigger model, more data, better performance?
Yeah, excellent point. Did that translate literally to the biological domain?
How big did they have to go?
Well, this is where it gets really impressive. The flagship model they talk about is C2S scale 27B. It's 27 billion parameters. It's a decoder-only transformer based on that Gemma 2 architecture we mentioned. And the pre-training data,
Mm-hmm.
it's enormous. Over 50 million individual single-cell transcriptomes, plus all the associated metadata and text corpora.
50 million. Wow. That's an unbelievable amount of data wrangling. Did they reach a point where the model just knew more detailed biology than any single person could?
It certainly seems that way. They tested models all the way from around 400 million parameters up to that huge 27 billion parameter one. And the key takeaway, the scaling laws held up perfectly. Bigger models consistently gave better, more accurate biological predictions and explanations. The performance improved predictably with size, just like we see with general language models. It's pretty compelling evidence that this translation to language actually works, that biological patterns follow these informational laws.
Okay, that's huge validation for the whole concept. But, internal metrics are one thing. How do they check if the model's outputs are really biologically meaningful? How do we know it's not just finding clever statistical patterns that wouldn't actually happen in a real cell? External quality control.
Right, and that's critical. They had to get creative here, move beyond just checking gene-by-gene accuracy. They needed to know if the overall structure of the generated cells looked real. The main metric they lean on is called the single-cell fresh inception distance, or scFID.
scFID. Okay, let's pause on that. For the listener, what is CFID actually measuring? Why is it better than just, say, checking if the gene expression numbers match up?
Think of CFID as a sophisticated check on biological geography. It measures the distance, or similarity, between two distributions of cells in some high-dimensional space. So it's not just looking at one cell, it's looking at the whole population. Does the shape and clustering of the cells the model generates match the shape and clustering of real cells? If you tell it to generate T cells, do those generated T cells end up in the same neighborhood in the embedding space as real T cells? It checks if the model understands the sort of geometric landscape of cell types and states.
And they used another model, scGPT, to create that embedding space, that map.
Correct. scGPT acts as the embedding backbone to calculate scFID. It's like using a trusted map maker to make sure your evaluation metric is reading the landscape correctly, respecting that single-cell geometry. But they didn't stop there. To really push the fidelity, especially for predictions.
That's the reinforcement learning part. The paper mentions group relative policy optimization, GROP. Sounds complicated. What does that actually do for a biologist using the model?
Yeah, GROP. It's essentially a fine-tuning step that uses rewards based on, well, biological common sense.
Yeah.
You're trying to steer the model away from generating impossible or biologically nonsensical cells. So for instance, if the model spits out a cell sentence that implies it's strongly expressing two genes known to be totally exclusive to different cell lineages, you give it a penalty, a negative reward. GROP helps align the model's predictions with known biological rules and even expected outcomes from lab assays. It's about making sure the model generalizes better, especially for tricky things like predicting drug effects it hasn't explicitly seen before. It forces the model's language to obey biological physics, if you will.
Okay, this is where it gets really exciting for me. They didn't just build a fancy model and test it on metrics. They tackled a real major clinical problem: immunotherapy. We know it often fails because many tumors are cold, right? They don't show enough flags antigens to get the immune system's attention. So the team used Cell2Sentence to hunt for a way to heat up those cold tumors.
Exactly. And the way they set up the screen was really clever. It wasn't just find any drug that boosts immune flags. It was much more nuanced. They specifically wanted to find drugs that would boost MHC-I antigen presentation, that's the main flag system, but only when there was already a little bit of an interferon signal present, just a modest amount.
What's so specific? Why the condition about the interferon? Why not just crank up MHC-I everywhere?
Because that would be like hitting the whole body with a sledgehammer. You'd likely get widespread inflammation, serious side effects. They were looking for something smarter. A context-aware amplifier. A drug that sees that little bit of existing interferon signal, maybe a sign the immune system is trying to respond, and then helps it along, amplifying the effect just where it's needed. You need a model that can understand the cell's state in condition A, baseline, predict the change with condition B, interferon, and then predict the outcome with A plus B, interferon plus drug. That requires real reasoning.
Okay, so they run this incredibly specific, context-dependent screen through the 27B model. What did it find? What was the novel prediction?
The model pointed to a drug called silmitasertib. It's a CK2 inhibitor. And crucially, the prediction showed exactly that context split they were looking for. A big predicted increase in antigen presentation when interferon was present. but almost no effect in the baseline neutral condition. That precise conditional effect, that was genuinely new, not something you'd readily pull out of the existing literature.
Wow. That specific conditional prediction really feels like proof the model isn't just pattern matching, it's actually reasoning about the biological text. But okay, prediction is one thing. The lab bench is the final judge. Did it work?
It did. This is the critical step. They took the prediction and tested it in the lab, in 2 different human neuroendocrine cell models. And importantly, these cell lines were completely new to the model. They weren't part of the training data at all, a true out-of-sample test. And the results, they confirmed the prediction beautifully. Adding low-dose interferon plus silmitasertib caused a significant jump in antigen presentation, much more than either interferon alone or the drug alone. That's the payoff right there. An LLM, speaking this language of ranked genes, generates a novel therapeutic idea, and it holds up in actual living cells.
So for you, the listener, thinking about maybe applying this isn't just some far-off theory. If you have your own single-cell data, how do you actually do it? How do you turn your counts into these sentences? What's the minimum recipe?
It's actually pretty straightforward, broken down into 3 main steps. Step 1, you got to normalize your raw counts. Standard procedure basically accounts for differences in how much sequencing you got from each cell. Makes it a fair comparison.
Okay, normalize counts. Step 2.
Step 2, rank the genes. Within each cell, you just order the genes based on those normalized expression levels, highest to lowest. You check out the actual numbers at this point. It's all about the relative hierarchy now.
Right. And then step 3, building the sentence. How many genes? There are thousands in a cell. How do you choose?
Yeah, good point. You need to manage the length of the transformer. So step 3 is take the top N genes from your ranked list. The authors often use like 200 or maybe a bit more, enough to capture the main biological signal. And you just convert that list into a string of text, gene names separated by spaces, highest expressed first. That text string, that's your cell sentence, ready for the model.
Okay, so you've got your sentence,
Huh.
but you can't just paste it into the model, right? Because it's trained bilingually. You need to talk to it.
Exactly. The real power is mixing the natural language and the cell data. So for example, if you wanted to predict the cell type, your prompt wouldn't just be the gene list. It'd start with something like, your task is to identify the most likely cell type for the cell described by the following gene expression ranking. Then you paste in your cell sentence. That kind of conversational query makes the big 27B model way more flexible and, frankly, interpretable than older models where you just fed in numbers.
So pulling this all together, Cell2Sentence feels like it offers a complete blueprint, doesn't it? It shows people how to actually use these huge foundation LLMs like Gemma for really complex biology.
It really does. It's a workflow. Step 1, encode your data into these robust cell sentences. Step 2, fine-tune the LLM, maybe using those biological rewards with GROP, focusing it on the specific biological question or assay you care about. And step 3, evaluate the output using a smart metric like CFID, making sure the results respect the underlying biology, the geometry of the cell states. It's a roadmap for making LLMs a practical tool in the lab.
So wrapping up, what's the big picture here? What does this all mean? It feels like Cell2Sentence gives biology this common language, a scalable way for biology and AI to talk to each other. It unifies gene expression data, all that complex metadata, even the scientific literature, into one system you can actually query. And the success, like with C2S scale model, it wasn't just about throwing compute power at it. It was about finding the right way to represent the data that ranked gene sentence and then proving that works where it counts, in actual human cells.
Yeah, and that brings up a really practical, immediate question for you, the listener, especially if you're working with this kind of data. The researchers themselves suggest the next step. So the final thought maybe to explore is this. Could you take a small, interesting subset of your own cells, convert them into sentences, and just prompt C2S scale? Ask it for explanations, the why behind what those cells are doing. Then maybe take the top, say, 2 or 3 explanations it gives you and design a quick focus experiment in the lab to test just those predictions. That's how you could potentially turn this paper, this deep dive, into a routine part of your own research process, from reading about it to doing it.